This program is brought to you by Emory University.
Thank you, everybody, for being here this morning. I have the pleasure of introducing Ray Hershberger. I've gotten to know Ray pretty well over the last 2 years. We've been a site for the precision medicine study. It's really been just an incredible opportunity to learn from you and gain a mentor and a friend. Most of you are probably familiar with Ray's work in the field of CV genetics. He has been at Ohio State since 2012, or the Ohio State, I guess I should say, since 2012. But this DCM project started way back when, I think when you were at Oregon. So he's going to tell us sort of about his journey and his thoughts about how to integrate CV genetics into our clinical practice.
That's great. Thank you. I'm going to set that there. Thanks, everybody. A nice rainy pre-holiday morning here in Atlanta. I'm delighted to be here, and thank you all for coming out on Monday. I am a heart failure, heart transplant cardiologist, for those of you who don't know me. I went to Ohio State in 2012 and decided to take a change in effort, and I directed a division of human genetics, so it's been a wonderful opportunity to have time beyond clinical care, of course, which takes so much time and effort. So I have these couple of small trials. They're not relevant to what we present today. I start this almost every talk with this slide. Genetics is all about phenotype and genotype, and the arrow reflects a bilateral communication. We have to know about both. Of course, phenotype is what we observe. That's clinical studies, medical testing, and of course, genotype now, it's sequence, and we sequence DNA, and that's laboratory studies, all kinds of molecular genetics to identify genetic cause. The phenotype we look at is shown in this very simple cartoon, and it's dilated cardiomyopathy, where that dilated left ventricle with contractile dysfunction, that has been the laser focus of this effort in cardiovascular genetics. But most of us learn from stories. Let me tell you a story. This is a real story. I do have permission from the family to talk about this family. In the middle there is a 14-year-old transplant recipient. And this is a successful story, at least in terms of post-transplant. The T-shirt says, yes, 1993. Some of you may have been actually maybe in middle school or whatever, but it was my first job at the Oregon Health and Science University. And David Newell, I met him one Monday. We had a Monday morning clinic. I got a call right after clinic from the pediatric cardiologist who'd picked up the service on a Monday. David had been in the pediatric intensive care unit at the Oregon Health and Science University. He was up on the 14th floor in its own unit for 2 weeks. And she said, I want to bring him to the transplant listing conference. We had our conference on Wednesday afternoon. And I want to talk about transplant because I just picked him up and he's been sick here for a while. He's not getting better. And after a couple of details, I said, let me send a fellow up. Let's look at him. Fellow called me. I went up to see him about 4 o'clock. He was gray, ashen, pale, actually had a blood pressure of 56, and was clearly acidotic, was in profound cardiogenic shock. And I actually did something I've seldom done in my career, only a couple of times. I asked for an amp of epi and gave him some mini boluses of epi to begin to resuscitate him, put him on high-dose milrinone, increased his inotropes. Called the coordinators, the transplant coordinators, and got him crash listed for transplant within 4 hours, record for the program. And amazingly, of course, this is back in the 1990s, didn't have VADs. They said they didn't have a bloom pump small enough. He looks pretty big here, but this is Kathy Crispell, a colleague and a fellow at OHSU, and the thoracic fellow. They were both short, and David was pretty small then. Couldn't get a balloon pump into him, so we were stuck back in the old days of medical therapy, of course, which we know very well. And within 36 hours, had a donor call and got him transplanted. Now, this is the success story. The ganciclovir is running. He was CMV high risk, and everything is wonderful. A year and a half later, I get a call from the University of Minnesota from a colleague saying, hey, what did David's heart show? And I said, why? He said, because his first cousin had sudden cardiac death. We're evaluating him at Minnesota for heart transplant. Well, in the meantime, we had started the Dilated Cardiomyopathy Research Project with the aims to identify and characterize genetic causes and then to translate that to the practice of cardiovascular medicine. And that's pretty much what we've been doing with this study forever. And so we called in David's mother and father and 2 older brothers. And lo and behold, David's father had been treated by his primary care physician with benzodiazepines for about 9 months for panic attacks at night. Well, when we saw him, of course, he had elevated neck veins and an EF of about 25% and a big liver and dependent edema, and was in having paroxysmal nocturnal dyspnea that had not been diagnosed. We actually diagnosed dilated cardiomyopathy in David's father at research screening. And that story, of course, is not unusual. I mean, Alana's helped, we'll get into this a bit more, but Alana's helping us with the study. And when you screen family members, it's amazing what you find when you look for it, but it's there. And actually, in our current study, it's about 10% of family members have DCM, don't know it. So the fly in the ointment was the first cousin was on mom's side of the family. Okay, so here's David. These 2 older brothers, I will go into this. We wouldn't make them black as in affected, but this was the first cousin here who had the sudden cardiac death. And I'll go through more of this family later on in the interest of time, but Dad was clearly affected, but Mom had to carry, on the mom side of the family, likely carried what we thought was a pathogenic mutation. And so we assumed that David probably was the unlucky son of his 2 older brothers, carried a nasty variant from both Mom and Dad. And so it was the 2 in combination that caused this trouble. Now, this was 1995. I'm an assistant professor. We're collecting preliminary data. Didn't get this thing funded till 1998, submitted grants in 97, and it took a while. I figured this is a one-off. You know, it happens, and it might be 1 in 100, 1 in 1,000, 1 in a million. I show you this today because this illustrates dilated cardiomyopathy genetics. And if you learn nothing else this morning, this is not simple Mendelian, but it's complicated. So it was unexpected complexity. It presented as simplex disease in an individual, but it was really familial. And then there was bilineal inheritance. How common is that? We still don't really know. What's a genetic model? The conventional would be autosomal dominant with incomplete penetrance. Penetrance is that genetic term that means if you have a disease-causing variant or mutation, the likelihood that it will actually present with some phenotype, something that we can see that causes disease. And then variable expressivity, variable age of onset, or multiple alleles with a multigenic model, or other oligogenic models, oligogenic meaning a few as opposed to monogenic meaning 1. And then, of course, I'm talking about very rare variants, things that are very rare, and I won't go into the math of this, but we're talking down in the range of 0.05 to 0.01% of population studies, not common variants, which are GWAS, but GWAS or common variants could be affecting the background to make this more evident. Of course, then there's something else, the environmental impact, and in general, we have not done a good job at all of really collecting environmental data. That's not completely true, but we haven't really integrated it in. Of course, it's a very exciting area, and I really won't touch on that. This is chemotherapy and alcohol and other things like that that clearly affect heart function. So what we study is dilated cardiomyopathy, defined as such. Of course, we sort out ischemics from non-ischemics. We do estimate that of the overall, if you consider this is actually, it's probably closer to 6 million with heart failure. If half of that is heart failure with reduced ejection fraction, and we estimate half or more is ischemic and the other half is non-ischemic, and then we sort out other causes as shown. What's left we call idiopathic. Of course, this is conventional cardiology. That's what we study. We do know that if you do family studies, that is, you do echo and ECG and quick history and exam on family members of someone who presents with IDC, you will find familial disease, and those have been single center studies, and the estimates range from 20 to 35%. The proportion of familial DCM, if you clearly have familial pedigree, so you're almost certain it's genetic, the sensitivity of testing is still only about 40% for dilated cardiomyopathy. So there's still a lot of stuff out there that we don't know. But this is certainly sufficient molecular testing for study and for clinical purposes, but it also emphasizes discovery. Further discovery is needed too. So it's still an enormous amount of work to be done. The problem is that most of what walks into our clinics, of course, appears to be simplex disease or one person that has dilated cardiomyopathy without apparent familial disease, even if you screen the family members. And the cause of that is still uncertain, certainly uncertain in the literature. And so we study it all, both familial and non-familial, to try to figure out this genetic background. The central hypothesis then is that DCM, whether familial or non-familial, has substantial genetic basis. And so this is what we study, and this is why we study it. Again, I'm a transplant cardiologist. I'm very familiar, along with Alana and Andy, and if there are other heart failure transplant people. This is where we work: drugs, devices, mechanical support, transplant. It works, it saves lives. It's even sometimes fun when we do fantastic things. But it's, of course, enormous impact and enormous cost, and it doesn't start with heart failure. It starts with dilated cardiomyopathy. But that takes months to years, and of course, in adults, we think that most of the time, based on our family studies, we know that a lot of these hearts are normal, and this dilated cardiomyopathy evolves over months or years. And the key point which we forget about is it is asymptomatic until it presents, and that presentation tends to be, of course, with fairly late phase disease. I mean, the heart's big, they're in heart failure, and they come in, and we're trying to rescue them and all of that. Now, there is clinical disease, even though it's asymptomatic, if you look for it, or there's risk of disease, and of course, the risk is genetic information, and genetics is all about one thing: risk and risk assessment. And ideally, we could then do surveillance echoes, and at the earliest sign of disease, come in with an ACE inhibitor or a beta blocker, or some other drug, or maybe even gene therapy, and prevent that. Or with clinical screening, if you find DCM, come in with drugs, conventional stuff. We know what to use to actually decrease the impact and to prevent this advanced phase disease. So this is, again, this is precision medicine. I'm not going to talk a lot about precision medicine per se, but this is a poster child. The channelopathies also can do this. The familial hypercholesterolemia people also do this. I mean, you find disease before you get to a myocardial infarction as an FH or something, or sudden cardiac death and long QT, and you try to intervene. Dilated cardiomyopathy, of course, we know drives enormous heart failure and a lot of morbidity, mortality, so it's particularly relevant. It's a little bit more complicated, though. So this is the classic types of cardiomyopathy. You know this. Dilated, hypertrophic, and arrhythmogenic. The HCM gene ontology is shown here. And basically, if you can find genetic cause, 80% of it is in two genes: MYH7, beta myosin heavy chain, or myosin binding protein C3, the myosin binding protein myosin binding protein C. Then if you add the troponins and tropomyosin, you get almost 98% of genetic cause. And as you know, hypertrophic cardiomyopathy is a genetic disease of sarcomeric proteins. And in general, hypertrophic cardiomyopathy is primarily a monogenic disease. There is some evidence of multiple mutations, but it's really 1 or 2 or 3%. And in general, hypertrophic cardiomyopathy, at least in a cardiovascular genetics clinic, which I do, my effort at Ohio State, I thought would be at least half heart failure transplant. It's morphed into 100% cardiovascular genetics. My clinic is booked out for 6 months, and everybody's on me because they can't get their patients in. But hypertrophic cardiomyopathy, pretty straightforward. Pretty straightforward. ARVC, these are proteins of the desmosome. Of course, the desmosome, a fascinating structure. I won't discuss that today. It's a talk unto itself. And ARVC is considered a genetic disease of desmosomal proteins important for cell adhesion and transcription and calcium homeostasis and gap junction function. In contrast, this is dilated cardiomyopathy gene ontology. And because this is Emory, and because it's Monday morning, and because you're all here, I'm going to go through each of these genes in exquisite detail. That's a joke. So the gene ontology is simply more complicated. The sarcomeric proteins, of course, we know those from hypertrophic cardiomyopathy. They also cause, variants in those cause dilated cardiomyopathy. Also cytoskeleton proteins, Z-disc, And then lamin, this nuclear envelope protein that would be on no one's short list, as well as the presenilins we contributed, phospholamban, of course, calcium handling, transcription factor, RBM20, a fascinating RNA binding protein, BAG3, a co-chaperone of heat shock proteins. Again, we contributed that to the literature. Mitochondrial ion channels, SCN5A. So it's a diverse gene ontology, which again underlies the complexity. This is a figure that we first put into review, and then it's in Brown-Wald's heart disease. I modified it, updated it slightly. Dilated cardiomyopathy is the center, but and all of these genes have some evidence in the hypertrophic cardiomyopathy. Now, a lot of these genes have minimal evidence to support it, but I was very inclusive here. The channelopathies kind of sitting down here by their own, although SCN5A does certainly cause dilated cardiomyopathy, some variants, but also a key channelopathy protein. But I just show this just to say that there's a lot of complexity here that has to be unraveled. So with this complexity, can we get to precision medicine? There are issues of genetic testing. I'm not going to go through. This is really more of I think of this as a research-oriented conference to stimulate your interest and enthusiasm for DCM research. Genetic testing, the sensitivity in familial DCM, 20 to 40%, non-familial, probably less. I will only mention the American College of Medical Genetics Standards 2015. That's kind of now the Bible of how you do genetic testing interpretation. They're more stringent, more conservative, so all of our testing standards have shifted in the last few years. And then the realities of DCM genetics, this locus heterogeneity, many genes I've shown you. Within those genes, the allelic heterogeneity, many different mutations across those genes can cause DCM, which just makes it complicated. And then this complexity, these unique or private mutations. So you might find something you've never seen before. There's no evidence out there. You call it a VUS because there hasn't been a previous report. There are guidelines. The Heart Failure Society of America had the first, this was published 2009, it was re-upped in 2018 in concert with the American College of Medical Genetics. It was a nice group, it took a lot of work, but important. This is a summary, a one-slide summary of the guidelines. Comprehensive family history always, family member clinical screening with a new diagnosis of any cardiomyopathy, refer as needed, especially syndromic disease or for children. I'm an adult cardiologist, and if it's syndromic, we punt to real geneticists or to those who are specialized in childhood disease. Genetic testing is recommended in the 2009 guidelines. We said should be considered. There's been a sea change, as you know, with genetic testing, and it's now commercially available, easily available, and it's actually excellent. Genetic counseling for all of the above. We do think that genetic counseling, patients understanding what's involved, both pre and post-test. And then this clinical screening is indicated with secondary findings. I won't comment on that, but it is new. The American College asked us to comment on that. That's when somebody does exome sequencing for cancer, and then they come across a cardiomyopathy gene, and then they come into your office and say, what do I do? And basically, you act like a cardiologist and take a history and exam and phenotype them. And if it's negative, there's some comments. If it's positive, then you would kick them into whatever the phenotype is and do the rest of it per guidelines. So, medical therapy, device therapy, and ICD therapy prior to EF, less than 35% in high-risk patients. That's if somebody has a nasty variant. You don't have to wait till their EF is less than 35 because they may not survive that long if it's a particularly pathogenic variant. Let's do a case. This is a real case, 58-year-old female, no prior cardiovascular history. She presented 2 years ago. You're a consultant now, and you're seeing this patient. She presented 2 years ago with bradycardia, junctional heart rate, long PR, atrial tach, sick sinus, has a pacemaker, negative angiogram, significant MR, but her LV size and function were okay then. So, and you're now consulted for second opinion because her EF is now down in the 40s, her MR is worse. She has new onset heart failure and considering a mitral valve repair. What should you do? Well, again, take a family history and phenotype the at-risk relatives if you can. This is her family history. She is the 58-year-old in black, and then a 63-year-old with a stroke at 50 with a pacemaker and implantable defibrillator, 66-year-old brother bradycardia pacemaker, implantable defibrillator, 71-year-old sister with bradycardia atrial fibrillation, pacemaker, and ICD. So this is, the EP guys, of course, are all over this one because this clustering of pacemakers and ICDs really suggests arrhythmic disease, except this woman has cardiomyopathy. And so family history is positive. Could she have a genetic cardiomyopathy? Yes. Is it relevant to help decide the surgical intervention? I don't know. I've been showing this case for 10 years, and I'm not going to pursue this question because it's complicated, particularly the MR replacement. But what cardiovascular knowledge could help inform this case? Family clustering of pacemakers with LMNA mutations is the most common. If you take unselected DCM, sort of 5 to 8%. If you enrich for conduction system disease plus DCM, it's probably upwards of maybe a third of cases will have LMNA mutations. And the guideline for genetic testing is recommended, so you test her, and at age 58, she did have a mitral valve repair. She didn't improve, but actually got worse, and she had progressive heart failure. I met her then at age 60. She was referred for transplant, was a great candidate, got transplanted and is doing well. What did we find? A LMNA mutation, as I have telegraphed, and unfortunately, she's in a research study. She was published in one of our papers. We didn't ever get DNA from the older family members, but we would infer that they likely were mutation carriers too. So molecular testing wasn't available when this patient underwent mitral valve replacement. I do think that if you take a family history, this could have resulted in a LMNA diagnosis, and again, the EP guys, at least at OSU, are all over these cases like that. And could have she avoided a mitral valve replacement? I don't really know that question. That's complicated, and that's well beyond genetics and cardiomyopathy to some extent. It's really a difficult thing, but at least you would have known that it may have added some additional risk that this heart may not get better with a genetic background. And there are, of course, I only mentioned gene therapy. It will happen. In fact, it is beginning to happen, and I hope it happens in my career. In fact, maybe it'll happen in Ohio State. Maybe it'll happen in the consortium, in the Dilated Cardiomyopathy Consortium. I'll say more about this. It will happen certainly in many of the younger generations' lifetimes in clinical practice. One could imagine, not this woman when her heart is end stage, it's too late. But one might imagine if we would detect this a little bit earlier phase and dial in a gene-specific therapy, it may actually avoid advanced therapies. That's the idea.
Right.
Yes?
Let people know we're doing that trial.
The LMNA,
Yeah, so we're recruiting for that.
the array trial?
Yeah.
Good. So Andy says, a shameless advertisement always for our friends at Emory. There is a clinical trial, that was in my disclosures. It's an effort to recruit LMNA patients that are sick enough but not too sick, that have an established LMNA variant, to throw in a p38 MAP kinase inhibitor for investigational therapy, and recruiting has been very difficult. But that's fantastic. I should have asked that ahead of time. So if DCM results principally from genetic cause, and most of it appears to be sporadic, then what are the genetic patterns of disease? So the usual patient that presents looks like this, where the black is affected and the white is unaffected, and the parents are alive and doing well, and the subject comes in and they have dilated cardiomyopathy, and we're saying that's genetic or heritable. Well, the possibilities are that it's a de novo mutation, started with the subject, but the parents don't have it. It's plausible. Or that there's reduced penetrance. One of the parents has this mutation that we think is causing disease, but for whatever reason we don't see it. The other possibility is that there is bilineal inheritance, and each of these mutations or variants are not penetrant, but the combination of them do cause DCM. And we would call that monogenic versus oligogenic disease. Most of the time, the family is more like this, where somebody comes in with disease. We test, we find something that is called a variant of unknown significance. We test the rest of the family, and we find 2 subjects who are unaffected. And then we do research testing, and we do exome sequence, and we find something else that also smells like it could be the real thing, and it could be bilineal. And then, of course, this black-white thing is almost never the case, and there's different varieties of shades of gray, and we have to sort that out. And that's effectively what we're doing right now in our studies. So what's the evidence that IDC, or simplex disease, has a genetic basis? This is published data. It was data from our Sanger sequencing studies that suggested that the non-familial frequency was similar to the familial frequency of what we thought could be possibly or likely causing variants. The caveat was that we didn't screen family members clinically. This was a single center study at Oregon, and we had families from all over the country that were referred in from colleagues. And we had a great family history taken, but few families were clinically screened, so we may have been missing those. So what's the evidence that DCM is oligogenic? This is a paper from Birgit Funke, a colleague who was at the Laboratory for Molecular Medicine, and basically they sequenced 766 DCM probands, and if you look at the frequency of variants of path, likely path, and high probability VUS, it was 7.3%. If you add in their VUSs, it was more like 44%, and then they had 319 cases with at least one of these. It was about 35%, so clearly multiple variants involved in some percentage, although that percentage was not very clear. Another paper, European group, 639 DCM patients, half sporadic, half familial. Just a comment, the data are not really provided, but 38% of patients have compound or combined mutations, 12.8% have 3 or more. So suggesting that there's something more here than just a single monogenic disease. These are unpublished data from our, what we call our legacy study. 175 familial pedigrees, 106 non-familial, 281 total, 412 subjects. And basically the 29.2% is simply adding the numbers of variants that we observe. This has not been published because, again, as I said earlier, the ACMG standards for variant adjudication have been changing. The goalposts have been moving, and we might eventually get those data published. But it's probably going to be somewhere in the 20s% range. We also published a recent paper, I won't perseverate on this, of our Lammen pedigrees, 5 families. Each also had 1 or more additional rare variants identified in a relevant DCM gene that were biologically relevant, and again, and there's statistical evidence in that showing that those variants were actually, again, biologically relevant. And then preliminary data from our precision medicine study using the same methods as that published paper. This is an initial cut, and I'll say more about the precision medicine study, but an initial cut of 114 restricted to, of probands with negative 37% VUS, 39%, a likely path 23%, pathogenic 4%, 114. And this is the breakdown of their likely path or path alleles versus VUS. So 37% had neither, none. 19% had 1 path or likely path. 27% had a VUS. This is not a common VUS. This is a very stringently defined VUS that, if we had additional data from additional subjects, may well be likely path or path. And then you can see the combinations here, 2, 5%, et cetera, and if you add it all up, basically about 20% of this current cohort, very preliminary data, looked like they had biologically relevant multiple variants. I'd say it's conservative. Then we looked at the family members. So the real question is, what's happening in the family members? And we restricted the cohort to age greater than 40 because almost all the patients were more than 40, and it was a very small data set. And the LVF in the probands, 25%, LV, EDD, Z-score, the standard deviation of how big the heart is, over 4. The family members, EF of 56%, LV, the Z-score, completely normal. And then the findings in the family members only shown here, basically the number of probands VUS alleles carried, the EF was decreased by almost 5 points. This is complex statistical genetics, and I'm sparing both me and you all of that background. The data from the Lammen publication, that the methods are essentially the same, and then also their hearts were larger with the Z-score, also statistically significant and larger. This is in a very small sample size, and we think that going forward, as we add in additional subjects from the precision medicine study, this will clearly play out. The key point is that in family members greater than 40 years carrying more of the LP or P or VUS variants, there was a worse LVEF and LVEN diastolic dimension, suggesting that those variants in those family members are biologically relevant. The VUS thing is particularly important because that has been a pervasive issue of what to do with those VUSs, and at least in DCM, we think that, we don't know how many of them, but certainly some of them are probably, I'm guessing the majority, but we don't have data yet, are actually biologically relevant. So the inference then is that they are biologically relevant for the development of DCM or related endophenotypes. Endophenotypes meaning EF or heart size or other kinds of more specific phenotype features. So might an oligogenic architecture explain in part what presents a simplex DCM? That's really the question here for us. And our family data illustrate that. I've told you the story of David. Let's come back to a real story. And again, I showed you this from 1999, this black and white pedigree, and basically this usual black and white approach is insufficient when you get to complicated, to beyond monogenic pedigrees. So we devised 5 categories for DCM study, ranging from 0 to 4, no cardiovascular disease, myocardial dysfunction, but not meeting formal DCM diagnostic criteria. This is grade 1. Asymptomatic DCM, symptomatic DCM, and then advanced DCM or DCM-related death. Those have been published last year. And then applying these criteria to David's pedigree. So this is David here. At 14, critically ill with heart failure, emergent heart transplant. His next older brother developed heart failure in his 20s, had a heart transplant at 31 years of age. This is at the Oregon Health and Science University. He was actually retransplanted. He's alive and doing well. Then the oldest brother, fully asymptomatic, the most he's ever had was borderline LV enlargement. Systolic function has always been normal. I told you about Dad, heart failure diagnosed at 44. He died of heart failure at 57, really didn't want a heart transplant. I took care of him at OHSU. And then Mom, who is still alive, still very active in the research program, was never symptomatic, we've echoed her probably 10 times over the last umpteen years, and sort of borderline LV enlargement, but no systolic dysfunction. And then the uncle, I didn't tell you this story, but at clinical screening had asymptomatic DCM, and a few years later had sudden cardiac death in church one Sunday morning, was resuscitated, as an ICD in place, and is doing well. Then this was the cousin who had the sudden cardiac death at 17, had DCM, but has actually been remarkably responsive to medical treatment and hasn't had any other disease, has an ICD in place, and is doing well. Okay, so I only put this in. We didn't find anything with Sanger sequencing, but I put this in for Alana, actually. I normally take it out, but this is the pain slide. This is Sanger sequencing. That's all we had for how many years? 15 plus years. We didn't find anything. And the reason that I'm still doing this is because we actually now have the tools to figure this out. And sometimes when it isn't the right time to get your stuff done, you just keep hanging in there until you finally get there. And then you keep accumulating data and accumulating families and eventually figure it out. So we moved to exome sequencing, and on the maternal side, we found an FLNC nonsense, a truncating variant, and that, I think, explains, that gene explains the maternal disease really perfectly. And its complete segregation, FLNC, is this actin cross-linking protein. It also has been seen in a skeletal myopathy, also hypertrophic cardiomyopathy, and then dilated cardiomyopathy, particularly with truncating variants. Very prominent ventricular arrhythmias with sudden cardiac death, which fits with this family, at least the dad and the son, and then relatively less dilatation and systolic dysfunction. So we think that FLNC actually explains the maternal side of the family. And again, I said most of this. Okay. So what about the rest of the family? Well, the paternal side, we found a Titan truncating variant. It's present here in Dad, oldest brother, older brother, and David. The Titan analysis, again, it's been difficult to assign pathogenicity to some Titan rare variants because, of course, only truncating variants are thought relevant for DCM. And in this pedigree, the Titan truncating variant we think is unlikely to be causal because of the lack of DCM in the older brother, the oldest brother. We do know that 1% to 3% of control samples of Titan truncating variants, and in our early experience, we found some non-segregation. There's no question that Titan truncating variants, and this whole subject of Titan, again, it's whole talk to itself, there is a fairly dramatic variable penetrance. And whether Titan at times acts as a risk allele or whether it actually acts as a pathogenic disease-causing monogenic allele probably varies. And again, we hope to define some of that in our current studies, but I think that's uncertain. So we identified this Titan truncating variant, and we don't think it explains all of the disease, but we think it does contribute probably. We also found a SOS1 variant, and we do think that this is in part where the money is. SOS1, for those I know, it's associated with syndromic cardiovascular disease, Noonan syndrome, which is generally pediatric, short stature, characteristic facies, generally hypertrophic cardiomyopathy, although DCM has been reported. Most of the time, it's PTPN11, and if that is negative, it's KRAS. If those are negative, then about 20% have SOS1 variants. And in this pedigree, the variant was, we found the SOS1 variant. It was absent in EXAC and NOMAD, and I don't go through this, but we just submitted a paper last Thursday to the American Journal of Human Genetics with extensive laboratory data showing that this variant, along with several others in the cohort, in the legacy cohort, actually are likely relevant for DCM. So we proposed the SOS1 variant explains why the oldest brother had minimal disease. He does not have the SOS1 variant. He only has the Titan negative for FLNC. The lack of the FLNC variant explains the later onset and more benign DCM in David's father. He has the Titan and the SOS1. The FLNC Titan SOS1 explains the greater disease in David and his next older brother. They have all three. But why did David have a much more malignant course? We also found a CAM kinase 2 delta variant that we think is a protective variant. The idea is that, you know, for cardiovascular disease, we always focus on nasty disease-causing variants, but we know that some variants, both common and rare, can be protective. And it turns out that CAM kinase 2D is a very well-defined protein. It's a serine-threonine protein. It's in the serine-threonine protein kinase family, and it interacts with phospholamban, of course, the key calcium-handling protein for cardiac excitation, contraction, coupling. It's actually been extensively used to dissect RBM20-induced disruptive splicing of key proteins, and there are models suggesting that if you decrease CAM kinase 2D, it is protective. So there's really excellent animal data to suggest this could be an operative mechanism. So we hypothesize, we don't have data yet, but Peter Moeller, who's a cell biologist at OSU, and they're trying to build a mouse for us, although they've had trouble getting this mouse to breed, and for reasons which are unclear. We do some zebrafish studies, and I told the guys that we should just put this into fish, even though it takes longer. But of course, the mouse guy's been working for over a year, and we don't have a mouse yet. So, I mean, we don't have data yet. So the idea is that if it's protective, Dad does have it. It helped him to live to 57. David's older brother had it, helped him to not need a transplant to age whatever it was in his 20s. But David didn't have it, so he had the full brunt of the Titan and the FLNC and the SOS1. So David's oldest brother didn't need it. Took me a while to figure this out. David's older brother was negative, but again, he doesn't have enough badness there genetically to really need that protective variant. At least that's the hypothesis. We do not have data, that is real data, that would meet scrutiny of a group like this to suggest that this is operative, but we hope to test that hypothesis. So in summary, this is kind of a rudimentary scoring system, and I just throw it up. David had the worst, and his next older brother had the next worst, and his oldest brother really had very little, and Dad had some, and that's the story. So it's not simple. It's complicated. It will get more complex. We have to figure out we're monogenic means oligogenic, and we have to create and test key hypotheses. Again, the central hypothesis, I've showed you this, and this is being tested in the DCM, what we call the precision medicine study. This is the study that Emory is a part of. It started enrollment in June of 2016 with 12 sites, and we needed more, and thankfully Emory joined, and with Andy's help, and then Alana's been the go-to person who's done a fantastic job. This is the active consortium sites in the precision medicine study. It's a wonderful group, heart failure transplant cardiologists. There's Emory with the circle around you guys. Thank you for helping. We're adding a couple of sites for family members only because we recruit family members all around the country. And then we're adding some sites for what we call the discovery study. I'll mention that at the end. And there are a bunch of sites considering joining the consortium to expand out both the diversity and the recruiting effort if we can get the discovery study funded. The issues in this study are the proportion of familial DCM, and our hypothesis is that about a third of it is familial. This will be the first single multi-site study to actually try to figure that out. How much of it actually is familial, so that when we're seeing somebody in clinic, if we actually screen family members, what the metrics should be that we should expect ourselves to attain. Issue number 2, cardiologists may not appreciate that familial DCM has a genetic basis, much less consider it for non-familial. And we suggest, based on our preliminary data, that whether it's familial or non-familial, it's pretty much all genetic. The third issue is the communicating risk from proband to family members has not been studied. Of course, the key thing is in the US where we have HIPAA, you just can't inform family members of somebody else's medical information, even if you know the family members, so that family members have to receive and understand risk information via the proband to act on it. And so we have devised, part of this study, a tailored intervention to help DCM probands communicate risk to their family members, and we think that will improve the outcome. Then the fourth issue we think is, again, very important. Idiopathic DCM has been suggested to be more common, morbid, and lethal in African ancestry compared to European ancestry, but we had, certainly when this grant went in, almost no data, no genotype, no sequencing data in African ancestry. It's been attributed to hypertension and other environmental causes. It was absent. So our hypothesis is that it's all the same whether you're white, black, or whatever. Just as a side comment, we do also think, and I will emphasize this, the analysis of diversity, of sequence from diverse backgrounds actually is extremely powerful to give us greater insight. So this diversity became evident in 2011 with the Exome sequencing project, and I happened to be a small part of that ESP, and it became crystal clear that we needed diversity to understand human, at least rare variant genetics. And of course, it does take extra time and effort, resources, and we all know that white guys are the easiest to recruit. And minorities have had historical reason for concern. We understand this. This is real. This is from 2009, this is GWAS data, 96% was European ancestry. By 2016, it was down to 81%, but still only less than 20% non-Northern European ancestry. But this is data from 2018, where non-European individuals had increased to 22%, but of that, African ancestry and Hispanics were only 3.7%. Remarkably, though, the non-European associations accounted for 46% of GWAS associations, a dramatic difference compared to what their contribution numerically was. And then of this, the African ancestry and Hispanic were 12%, contributed 12% even though they only were less than 4% numerically in contributing to, and this again is GWAS findings. So our aims were to conduct our aims are to conduct cardiovascular phenotyping of 1,300 probands, balanced between European and African ancestry. We sequence them, we identify genetic cause, and we evaluate an intervention to help communication of family-based risk. Again, in a graphic, this is a classic genetic study. We get phenotype clinical information, sequence with exome, do exome sequencing, return genetic results in a randomized study to assess this intervention. And this intervention is done at baseline. At proband enrollment, we randomize 1-to-1 to a communication tool. I will show you that in the next slide. Actually, I'll show you, yeah. And this is what we call Family Heart Talk. It's a booklet that was devised to explain how DCM is inherited, what family history is, a bit about genetics and how it plays out in the population, and some family pedigrees and what should the family do and how to talk with your family and all of that. So we randomize patients, give on a 50-50 basis stratified by site and race, and then we do family member recruitment. The first endpoint is the number of family members that actually come into the study and get clinical screening completed. We then return molecular genetic results, and then a second endpoint is family member adherence to the recommendations in the letters that go out to the family members of what they should do. That's our website. We had a Hispanic supplement from the Genome Institute, so it turns to Spanish if you need Spanish language. And I'm very pleased to say that our European ancestry enrollment has completed at 600. Our Hispanics were 100. Our African ancestry is at 475. It's really important. And we decided, in consultation with our Observation Study Monitoring Board, that's the NHLBI Oversight Group, last summer to persist here and extend our enrollment for another year to finish this off. Statistically, we're really on the knife edge of power. We really need 600 white and black to really do what we need to do in this study statistically. So, our family members, we have almost 1,800 family members enrolled, about 75%. We're very pleased with that. We continue to enroll family members through the end of the study. And let me finish up here with kind of a summary of where we are. Our current study here, active enrollment through 2020. We are very pleased that we have an ancillary study through an ancillary RFA funded. It is to collect family member, to collect echo data from all subjects. Centrally, Central Northwestern, they will do speckle tracking analysis and do strain imaging of these family members to detect an early phenotype. As some of you may or may not know, Alana submitted a metabolomics R01 as an A0, and hopefully that will be able to get funded. That would be, I haven't mentioned, but all of the probands and affected family members, we collect not only blood for DNA, we also collect lymphocytes for future possible iPS stem cells. We're not funded for that. We also collect serum and plasma, and of course the plasma is extremely valuable for additional studies. We haven't been collecting plasma in family members, but we have implemented that at OSU with Alana's interest in this, and with the idea in this grant to collect plasma in all family members going forward. So we need to complete the study, and then additional studies really of how genetic risk plays out in these family members. We've just received word of this CMR imaging study, and we don't have a follow-on echo study for repeat measures in the family members that really ought to be funded. We're very pleased that this echo R01 was activated this past summer, and then we just received a review of the CMR study, and that would be to take family members forward. Emory is a part of that study. We took the 9 sites that were the highest enrollers that had Siemens equipment, and you guys were in it. We're very pleased about that, and that will be starting. We don't have a notice of award yet, but we think that will be starting this summer. So now I proposed to figure out this complexity, we need numbers. You know, the GWAS people are talking hundreds of thousands now in these large data sets. We also need large numbers. For rare variants, a large number would be 10,000. And right now, that goal is aspirational. We have no funding for it, but we've been talking about it I've been talking about it really for at least 2 or 3 years. And the idea is that when we complete enrollment here to move sites to this new study. We'll see if it'll ever happen. All I know is you can't get something unless you plan it and ask for it. May not happen, but that's the goal. We absolutely scientifically need this to decipher the complexity issue. And so how are we going to do that? We've been preparing for this. We have a single or central IRB, which is essential. We're expanding the consortium. We're developing a study and getting preliminary data and talking to the NHLBI. We're doing a direct-to-consumer build-out, which I haven't only mentioned, and we're just bulking up. And then we also need to publish papers. I don't mention that here, but we have a wonderful publication and presentation committee. And just to say, if there are people at Emory who are academically inclined in cardiovascular genetics, this is an enormous wealth of opportunity to do additional hypotheses that may not necessarily be the ones that the study investigators would do ourselves, but feel free to reach out. So in summary, we understand Mendelian aspects of DCM genetics. It's clearly ready to be deployed in the clinic, but to truly attain precision medicine, I do think we need to understand DCM complexity beyond Mendelian. We need to define the boundaries of that, and then the way forward, of course, much larger cohorts, very well phenotyped and sequenced, robust analysis, and then functional studies, which I've only just mentioned very briefly. But this really requires a whole community of scientists across the spectrum, from clinical to really very basic scientists, to figure all this out. I'll just mention the site consortium principal investigators, recognizing all of them. Gordon Huggins is serving as a co-PI at Tufts. There's the next slide, and there's, again, Emory emphasized again. Our collaborating investigators, et cetera. Our clinical research team at OSU, a great group. We have an extensive informatics computational team with the build-out at the Ohio Supercomputer Center, which, again, has been extremely helpful, and other people. And then let's see, I'm missing a picture. It's not coming up. Anyway, thank you for the opportunity to present, and I'm delighted to take any questions.
Thanks for a great talk. So, will you, I guess maybe you need the larger study, but at what point will we be able to use this to help us understand the differential responses to therapy?
Yeah.
Because some of these, as we all know, some of these patients do great. Sometimes their EF gets better, sometimes it doesn't, but there's difference in their phenotype in terms of their responses to therapy.
Yeah, so the question, I guess everybody could hear it, so is what about therapy? And I'm focused on genetics and mechanisms, even though I'm not really a basic scientist, but I don't know when we will be able to when we'll have enough data to really figure out therapeutic responses. One of the issues, at least with dilated cardiomyopathy, is the heterogeneity, both the locus heterogeneity, these multiple genes. And so one of the issues with all of us in this LMNA study is finding enough patients that are at the right phase of disease to actually test an intervention. So that's one of the reasons. It's kind of the backside of proposing a very large cohort, so we could actually imagine having 20 or 30 or 50 or maybe 100 LMN patients, and maybe their family members, at different stages of disease, to actually figure out differential responses to therapy. That's number one. Number two, the sensitivity of testing. While ECHO is great for screening family members, I think to do real research we need CMR. And that's why we've been trying to get the CMR thing funded for, a couple years, and finally got it funded now, or going to get it funded. So I think we need CMR with all of that. Again, I'm not a CMR person, but the T1 and T2 and gadolinium and then the strain and extremely sensitive measures so that we can detect early disease, even define what we call a pre-DCM phenotype, to be able to get at some of these issues. But it's not a very pleasant answer. I guess the other thing is, while drug therapy can go forward in terms of doing the kinds of studies that that pharma usually funds, the large studies, I'm not sure there's enough return on investment for pharma to ever do this. then you're talking to biotech, and you're really thinking gene therapy or gene-specific therapies, so it's a conundrum. And in fact, at a group like this type of, you get it, and I think, I'm not sure what the way forward really is. And at what point we can sort of reach that tipping point to really move into gene-specific outcomes with, regardless of what the therapy is. Sorry for the long answer, it's complicated. So.
Just a curiosity that I've had over the years in seeing patients where family members had cardiomyopathy was they seem to be more likely to have higher heart rates. And those that did have higher heart rates or that later went on beta blocker therapy might have improved. I've had patients where they then stopped their beta blocker after improving, got worse. And just this past week, I had a patient who has a known Titan mutation. His son has it as well. He's affected. He's had a cardiomyopathy for about 10 years and has been on beta blocker for some time. And after going on ivabradine, he was on 50 of carvedilol twice a day. Going on ivabradine, his EF has now gone from 20 to 45.
Wow.
And so just as far as the phenotype, have you looked at in the screening and these unaffected or the affected family members at what their resting heart rates are?
Yeah, the short answer is no, but
yeah.
it's a paper. You can write it.
Yeah.
I mean,
do you think there's anything to
I don't
the
know.
concept?
Again, I'm like you. I'm a real clinician. You see these things,
Yeah.
and I think, and that's why I have always insisted that we have real heart failure transplant cardiologists doing our patient, acquire a patient so we can actually get authentic, real phenotyping. And I think that there are multiple hypotheses, but I'm at the point of show me the data. And we're trying to accumulate this data, and then again, a testable hypothesis, and then in a rigorous way try to prove it or disprove it. We just haven't. It's kind of a burr under my saddle. The cancer guys have been doing genetics, and it's all over. I mean, and in some respects, they're way ahead of us. And I run a division of human genetics, and I mean, obviously, the genetic counselors and cancer people, I mean, they're all over it, and it's fantastic. We just don't have the data. We just don't have the data. We've got to get the data. We have to have large enough numbers, large enough data sets to actually figure it out, and I think all of this stuff will come out. Thank you very much for the opportunity to participate.
Could I ask you one more?
Yeah, sure.
So I'm just curious, in doing all this genomic research, are you studying only the affected families? Is there any collection? In other words, are these variants existing in the general population? Is there any way to figure that out?
Yeah, yes, there is. And I haven't, I didn't provide all the background methods and stuff. We do have a design paper published, but the short story is we use now very large databases. NOMAD is the name of this huge database that now has closing in on 200,000 different either exomes or genomes that's in a publicly accessible database. So we have, and it's defined by ethnicity and race, so we can drill down. So all of this stuff that I'm showing is either extremely rare, down to 0.0001 or less in terms of frequency, or unique, never been seen before. So as opposed to common variant variation GWAS stuff, which by definition generally is greater than 5% of the population. So yeah, this stuff is very rare. Now, rarity does not necessarily mean that it's pathogenic. But the idea goes that if you have DCM and if you estimate it's 1 in 250 in the population, which is probably a conservative estimate, then if something is very common in the population that we think is relevant, then DCM should be 1 in 10 or 1 in 50. So that's why this frequency of variant in the population is critical, and that's all been integrated into our analyses. It's complicated, but yeah. Yes?
Did David have a left bundle branch block?
You say, do they?
Did David, your first
No.
patient?
No.
Do you think the EKG is of any value in
Oh, yeah.
screening,
Oh, yeah. Oh, yeah, for sure.
looking for?
Oh, yeah, absolutely. We haven't, in fact, that conversation came up with Gary Haas, who's a wonderful heart failure transplant cardiologist, a senior guy at OSU, and I got 20% of Gary's effort to help with all our phenotyping. Every case that comes in, we make certain that it qualifies, and if not, it gets kicked out, and Gary does that. We're having a talk exactly about this, about ECG, and in fact, Gary has gone through and has validated all of our database for ECG data. We haven't looked at that at all, but that report, that pedigree I showed you from 1999, Jack, there's another family in there where another family we screened. A woman had a big fat left bundle branch block. 5 years later had sudden cardiac death with DCM. And at her screening, initially, the only thing she had was that left bundle. And her LV systolic function and heart size were normal. Now, we don't have, again, this comes back to the gene-specific data question. Was this a one-off? But I think that electrocardiographic abnormalities may well be relevant. But in terms of, again, a systematic, rigorous evaluation in a large data set, gene-specific, lamin has been the only thing. There's a fair amount of publications of lamin because it's relatively common in DCM, and because of the electrophysiological phenotypes, there's been a fair amount of publication. Most of that's been single-center stuff and then accumulated over years. A cross-sectional study like this, I just haven't done it, but I think it's very important. Yeah. I was going to say, I can answer questions here all morning, but I know you have a lot of work to do. And again, I thank you all for the opportunity to come to Emory. It's delighted to be here. So,
Here's my question.
uh-oh.
Do you think you'll ever be able to retire at some point? Because
Well,
it
a good
seems
question.
like it's all getting very, very exciting, and it's cool to see all the work that you've
Yeah.
done for so many years.
Well, you
with
see that
all this.
timeline starts at 2013 and goes to 2029. So, my goal is to get this discovery study funded and get the rest of the stuff and get people like Alana funded, the next generation funded, and then I'm going fishing. So.
The preceding program is copyrighted by Emory University.